Bismillah ar-Rahman ar-Rahim. This is the second part of the cytology data show, and in this picture you can see the diagram for the ribosome formed of a small subunit and a large subunit. In between these two subunits we have the messenger RNA, and these sites are one for the addition of new amino acids and this for the polypeptide chain, P site and A site. Here you can see this small subunit and large subunit, and between we have the messenger RNA, and this is the third type of the RNA, which is a transfer RNA. At one end it is joining an amino acid, and in the other end it has the anticode, which is pairing with certain sequence of nitrogenous bases in the messenger RNA. As the ribosome moves along the messenger RNA, translation takes place with the formation of the polypeptide chain. This is like microscope picture showing the nerve cell, which is characterized by being branched cell, giving rise to the dendrites and to the axon. Here we have in the body itself, we have these basophilic parts which are called Nessel granules. These Nessel granules represent the ribosomes and polyribosomes present in the cytoplasm. And this basophilia is called focal basophilia. As we all know that the basophilia are of three types: diffuse, where there is a diffuse basophilia, and this is in the embryonic cell, which is very active in protein synthesis. Localized basophilia, which can be seen in the base of the cells forming the pancreatic acinus. And focal, like this in the nerve cell, where these foci of the Nessel granules or the ribosomes and polyribosomes. So these are the ribosomes or polyribosomes that can be seen in the cell body as well as in the dendrites of the nerve cell. You have to know and to study carefully the properties of these ribosomes. They are non-membranous organelles, and they are formed of the small and the large. In the middle of the large subunit we have the emerging specific polypeptide chain, and in between we have the messenger RNA. Here we have two types of the ribosomes. We have the free ribosomes and the attached ribosomes. The free ribosomes are single or in group called polyribosomes or polysomes that are formed of many ribosomes joined together by the messenger RNA, and they appear as a rosette like this one or spiral form. And the attached, which is attached to a certain receptor on the rough endoplasmic reticulum called ribophorin. Phorin means carrier for the ribosome. Also, you should remember the size of the ribosomes. It is from 20 to 30 nanometer, as well as the function, which is protein synthesis. For the free ribosomes, the protein synthesized is used by the cell, inside the cell, while the attached ribosomes form a protein that will be exported to the outside by exocytosis, as in the pancreatic acini producing pancreatic enzymes, as well as in the cells of endocrine glands producing protein hormones. This is the other part of the non-membranous organelles, which is called the cytoskeleton. The cytoskeleton is formed of microtubules and filaments. The microtubules form centrioles, cilia, and flagella. The centrioles are formed of two cylinders perpendicular on each other, and they are surrounded by a matrix rich in tubulin, which is a protein. This structure as a whole is called centrosome. Zome means a body, which is formed of these two cylinders of centrioles formed of microtubules and surrounded by a matrix rich in tubulin. In the electron microscope picture, you will notice that the two cylinders, one of them will appear in transverse section and the other will appear in longitudinal section as they are perpendicular on each other. Here we have a diagram showing the structure of LS longitudinal section of a centriole. You will notice that it is formed of 9 triplets because each group is formed of 3 microtubules. The innermost is the A, the middle is the B, and the outermost is the C. And here in the transverse section, the same is present. Here we have the 9 triplets, and the inner one is the A, the middle is the B, and the outermost is the C. Again, this is an electron microscope picture for the transverse section of the centriole. Here we have the 9 triplets, and this is the arrangement of the microtubules that form this triplet. This is the A, which is a complete microtubule formed of 13 protofilaments, while the B is sharing 3 with the A, and it is formed of 10 protofilaments, and the same for the C, which is the outermost. This is a diagram to demonstrate what I have said now. This is the A, the innermost, the B is the middle, and the C is the outermost in the triplet. The A is formed of 13 protofilaments, B is sharing 3 with A, so the remaining is 10, and the same for the C, sharing 3 with B, and the remaining are 10 protofilaments. So the microtubules are functioning in formation of the centrioles, cilia, and flagella. This is an electron microscope picture showing a transverse section in the cilium. The cilia is formed of 3 parts. We have this basal body, which is exactly like the centriole, 9 triplets. Here we have, after the basal body is present under the cell membrane, and it results from the division of the centrioles, the microtubules A as well as B will grow upwards, while C will grow downwards. The A and B will grow upwards in each triplet to form 9 doublets, and 2 singlets in the center will form these 2 singlets. Why they are singlets? Because they are not sharing the microtubules with each other. They are separate from each other. Here we have 2 singlets and 9 doublets. So we have 9 by 2, 18, and then 19, 20. So we have 20 microtubules. And notice that the cell membrane covers this extension. This extension is called shaft or axoneme, while this is the basal body. There is a difference in the number of the microtubules, and also the axoneme is covered by the cell membrane. So in this picture, this is a section in the shaft, as we have 2 singlets and 9 doublets, and we have this is the cell membrane covering the microtubules. Don't forget that we have a third part, which is arising from the downward growth of the C microtubules that will be discussed shortly. This is an electron microscope picture demonstrating the longitudinal section here in the cilia, and here you can notice the rootlets arising from the downward growth of the C microtubules in each of the 9 triplets. And here we have the microtubules of the shaft, and this is the cell membrane covering the shaft or the axoneme. So these are the cilia. This is a cell membrane of this cell, and this is a cell membrane of the other, and this is the junctions between the 2 cell membranes. We have here the tight junction and adherent junction. In this picture, we have the EM of the microtubules and the actin filaments or the microfilaments. Here you can see these are the cylinders of the microtubules. They are very evident. And these are the fine threads of the actin. The actin here, and this is the microtubules. Notice that the microtubules as well as the actin will help in the intracellular transport of any particles from one spot of the cell to another. Don't also forget the size of the actin is from 5 to 7 nanometer, while of the microtubules is 24 nanometer. And also don't forget that the actin is the microfilaments. We have also intermediate filaments which have a diameter equal about 8 to 10 nanometer. So you should remember these sizes: the 24 nanometer for the microtubules, the 5 to 7 nanometer for the actin, and the 8 to 10 nanometer for the intermediate filaments. Also, don't forget the structure of the microtubules. It is formed of tubulin, and the actin is formed of 2 chains of G-actin, which is globular actin. This is a chain, and this is another chain. I will draw in a different color of also the G-actin, and by this way they are twisted over each other to form the F-actin. So each chain is called G-actin, and the two will form F or filamentous actin. How to study these sizes? Here we have the actin is 5 to 7 nanometer. 5 to 7 together are 12 nanometer multiplied by 2 equal 24 nanometer, which is for the microtubules. The intermediate filaments, you will add another 2 for the size of the actin. So for the intermediate, I have from 8 to 10 nanometer. This is a diagram demonstrating the important function of the actin or the microfilaments, which is that it is present in the microvilli. The microvilli are present on the surface of absorbing cell like the intestinal cell to increase surface area of absorption. They are supported by these actin filaments or the microfilaments. Also, the actin is present in the base of the the actin filaments present in the microvilli. We have at the base we have another actin. This actin present at the base is called terminal web. Terminal web. This is a part which supports the actin vertically arranged in the microvilli. Notice that the actin here will help to change the length of the microvilli. It can cause elongation and shortening. And notice also under these microfilaments we have intermediate filaments here, which are thicker. As we have said, it is from 8 to 10 nanometer, while the actin is from 5 to 7 nanometer. This is an electron microscope picture demonstrating the microvilli. The microvilli, they are projections on the surface of the cell membrane, Here we have, this is a cell membrane covering the microvillus. And you will notice the two dark lines with the electron-lucent area in between, characteristic for the appearance of the cell membrane. Here inside the microvillus, you cannot see microtubules like the cilia, but you will see these fine threads of the actin or microfilaments. This is a junction between this cell and the other cell. So this is a cell A and this is a cell B, and these are the junctions between the two cell membranes. On the right side, you can see the transverse section of these microvilli. And inside it, you can see these dots representing the threads of actin transversely cut. And the most important is the presence of this cell membrane. And over the cell membrane, you have this cell coat. So we have cell coat, cell membrane, and inside we have these dots and not microtubules. So this is the microvillus. What is the difference between this microvillus and the shaft of the cilia? The difference appears in the center. If you look and you find dots, so these scattered, so this is a microvillus. And if you see 9 doublets with 2 singlets, it is a cilium or a shaft section. This diagram summarizes the functions of the actin. It is present in the microvilli and it helps in elongation or shortening, as you see. This area is the terminal web. Also, it is present under the cell membrane in this area, which is called cleavage furrow, that appears during the separation of 2 daughter cells in the last stage of mitosis or meiosis, where the 2 cells should be separated. Here you will have this depression, which is cleavage furrow, and inside the cytoplasm, under the cell membrane, the actin together with myosin. You see here thick line and thin line. The thin is the actin, the thick is the myosin. So in this area, the myosin is sharing in the division of these 2 cells from each other. Also, it is present with myosin in the muscle. It is present under the surface of the cell membrane to form a network that help in changing the shape of the cell, as in forming pseudopodia and helping in the movement of the cell as a whole. Don't also forget that their presence inside help for the movement of any particles from one side to another. As regards the microtubules, their functions is the formation of the cytoskeleton, which help to support and maintain the shape of the cell. Number 2, it enters in the formation of the cilia, centrioles before the cilia, centrioles, cilia and flagella. Number 3, it is important for the formation of the mitotic spindle during the cell division. Also, number 4, it helps in the transport of particles from one side to another inside the cytoplasm of the cell. This is a light microscope picture for the stored food as an example for inclusions. You should remember the characters of the organelles and the inclusions. Of course, the inclusions are non-living, temporary, not present in all the cells. They are not vital or essential, and they represent the metabolism products in the cell. Here we have the light microscope picture for the stored glycogen, which is always prominent in the liver cells. On the right side, we have these glycogen granules are stained red by best carmine. Best S, carmine. And on the left side, it is stained with the PAS. Of course, you notice the color here is red, but here it is purple, or what is called magenta red. These are the liver cells. This is the nucleus, and these dots represent the stored food. This is a blood sinusoid, of course, between the liver cells. On the right side, also it is the blood sinusoid here, and these are the liver cells, and this is the nucleus of the liver cell. These dots are representing the carbohydrate molecules stained red by Best carmine. This is an electron microscope picture demonstrating here the glycogen granules aggregated together to form what is called beta granules. What other type of granules that appear in the electron microscope for the stored glycogen? It is the alpha, which are single granules, but the beta are aggregated granules. And of course, you see all these electron dense granules grouped together and aggregated to form these areas. These are the beta granules for the carbohydrates stored in liver. And this is, of course, the rough endoplasmic reticulum with the attached ribosomes, parallel flattened tubules in this picture. If you are asked what is the organelle that is responsible for the utilization of glucose originating from this glycogen breakdown in the liver cell, the answer will be smooth endoplasmic reticulum. This is a light microscope picture for another example of stored food inside the cell. As an inclusion, it is the picture of the fat stored in the fat cells, but here it is stained by hematoxylin and eosine. Here, due to the dissolving of this fat inside the cytoplasm during the preparation of the section, the fat will appear as empty white spaces. So this is the cell here, and this is the peripheral flattened nucleus, and this is the small rim of cytoplasm. The most important here is this big fat droplets present inside each cell. And this is, of course, a unilocular cell. But if you want to see or stain this fat, you should apply a special stain which is called or Sudan 3, that will stain the fat by an orange color. Also, you can use Sudan black or osmic acid that will stain the fat in the cell by black color. And this is, of course, a picture for the white adipose connective tissue, where we have the unilocular fat cells, and these are bands of the collagen fibers, bundles of collagen fibers. This is a light microscope picture for the nuclei. This is the nucleus, and this is the cell. As you see, the nucleus may be rounded, may be oval, and different types of shapes, like the segmented, like the lobed, like the kidney shaped, like the cartwheel, and also they differ in their position. They may be central, they may be peripheral, they may be eccentric. And here we can see that inside the nucleus we have this dark area which representing the nucleoli. The cells may be binucleated. This is an electron microscope picture demonstrating the structure of the nucleus. Here we have, first thing is the nuclear envelope, formed of outer layer studded with the ribosomes, continuous with the rough endoplasmic reticulum. The inner layer, which is the fibrous layer, it is related to the chromatin threads. Don't forget that the outer nuclear membrane is called the granular, or it is characterized by being granular, and the inner is characterized by being fibrous. Also very important to remember that here we have an intermediate filament related to the inner nuclear envelope and responsible for its support. This is called lamines. Then we have another feature also related to the nuclear envelope, which is the nuclear pores, at which the outer and the inner are joined together. These pores are covered by octagonal annulus or ring. What is the function of this nuclear envelope? It is the control of transport of substances from the cytoplasm to the nucleus and from the nucleus to the cytoplasm. Mention one material that will pass from the nucleus to the cytoplasm. Of course, you will remember the ribosomes and the RNA. And mention one substance that passes from the cytoplasm to the nucleus. It is the histone, as well as the protein needed for the formation of the ribosomes. Here we have the heterochromatin, which is darkly stained, and according to its location, it is the peripheral heterochromatin. Of course, you know that it is the coiled inactive part of the chromatin, and then we have lightly stained dots here. This is the euchromatin, which is the active because it is the extended part of the chromatin network. This is also heterochromatin, present as islands between the peripheral and the nucleolus. The white area here between the dots, it is the sap. This is the nucleolus. Here we have this organizer in which we have the DNA from which the ribosomal RNA is formed. And then this part is the fibrous form of the newly formed ribosomes, so it is called pars fibrosa. And this part is the granular form of the ribosomes, so it is called pars granulosa. And these areas which are lightly stained, it is the sap. All of this spongy structure, not surrounded by a membrane, it is called the nucleolus, and it is responsible for the formation of the ribosomal RNA. Again, this is the pars amorpha or nucleolar organizer. This is the pars fibrosa immediately surrounding the nucleolar organizer, and this is the pars granulosa, and this is the light areas where the sap is present. Don't forget that the pars fibrosa, the fibrosa, together with the granulosa are forming what is called here nucleolonema. Nucleolonema. So if you are asked what is the nucleolonema, it is the pars fibrosa and pars granulosa in the nucleolus, and we know what is the difference between fibrosa and granulosa. Again, this is an electron microscope picture for the nucleus, but this nucleus is characterized by having abundant euchromatin, more than the heterochromatin, and this denotes that it is an active nucleus, and it is called euchromatic nucleus. Euchromatic nucleus will be present in any active cell, like, for an example, liver cell. These are the mitochondria, and this is the euchromatic nucleus. Here we have the nuclear envelope. Here, this is the peripheral heterochromatin, and this is the chromatin islands, and this is the nucleolus associated heterochromatin. And this area, it is the nucleolus, of course, and here we have amorpha, and then the fibrosa and granulosa are not so evident in this picture. Here we have also, this is the heterochromatin nucleolus associated, all these dark areas, and all of this is the nucleolus. This is also nucleolus associated. The lightly stained dots, these are the euchromatin. This is a picture which is characterized by having a nucleus with the abundant darkly stained chromatin. So here we have the heterochromatin is very prominent, so denoting that it is inactive nucleus. And also you may observe that the nucleolus is not observed, so it is a typical heterochromatic nucleus present in less active cells. Of course, these are the mitochondria, as you see in this picture, this one and this one, and they are very minimal in this cell. Give an example for a heterochromatic nucleus. It is the nucleus of the small lymphocyte. This picture demonstrating the nuclear envelope, outer granular and inner fibrous, and this is the peripheral heterochromatin, and this is the euchromatin. And this is the nuclear pore complex. Here we have the union between the outer and the inner, the outer and the inner, the outer and the inner, and this area which is called the nuclear pore complex. It is circular and it is occupied by 30 nucleoporin proteins that form a ring. So this ring is formed of here 30 nucleoprotein forming a ring or an annulus called octagonal annulus. Here the nucleoporin filaments, you should know that nucleoporin filaments extend into the cytoplasm and the nucleus. هنا we have filaments that extend to the nucleus and to the cytoplasm. If you have a label in this area, it is the annulus or pore or ring, nuclear pore أو octagonal annulus or octagonal ring. And of course, you know the function of these pores as we have said before. Also, I want to add that this space between the outer and the inner is called perinuclear space. It is from 30 to 50 nanometer. So again, I will revise the different dimensions. The actin from 5 to 7, the intermediate from 8 to 10, the microtubules 5 and 7 will be 12 multiplied by 2, so the microtubules will be 24 nanometer. Here we have also the ribosomes. The size of the ribosomes is from 20 to 30. Here the perinuclear space is from 30 to 50. This is an electron microscope picture demonstrating this cell. This cell is characterized by having a lot of rough endoplasmic reticulum, as you see, highly extended in the cell. And also we have mitochondria. This nucleus here, you demonstrate this heterochromatin in the peripheral position, and we have nucleolus associated, and we have islands. The appearance of this nucleus is very characteristic. It looks like a clock face or a cartwheel appearance. Also notice here the outer granular layer of the nuclear envelope, very evident. This appearance is very characteristic for the plasma cell, which is characterized by having a cartwheel appearance or the bands of heterochromatin alternating with the euchromatin. And of course you can see the nucleolus very evident, so it is not inactive nucleus. Now let's start to assess our knowledge. The red arrows here point to rough ER. Green arrows point to mitochondrion. Name a stain, iron hematoxin, or Janus green, and one visible feature, the presence of the folds or shelves or cristae projecting to the inside. Green arrows point to the cell membranes of two adjacent cells. The red arrow points to the intercellular space. The star lies in the cytoplasm. Here, this is a cell membrane, trilamellar appearance, and this is another cell membrane, trilamellar or unit membrane appearance, and this is the space in between the two cells. The molecules in the rectangular area, phospholipid bilayer or phospholipid molecules. Yellow arrows point to integral transmembrane protein. Blue arrows points to the glycogen of the glycolipid molecule of the coat. If you have a label here, it is glycogen of the glycoprotein in the glycocalyx, which is the other name for the coat. The organelle is called mitochondria. Blue arrows point to shelves or cristae. Arrowhead points to matrix of the mitochondria. Name structure is pointed by red arrow. It is the Golgi apparatus. Green arrow points to the secretory vesicles. Yellow arrow points to transfer vesicles. This is the concave, the mature. It is the trans phase, so this is the secretory. These are flattened and having this appearance very near to each other, compressed, and this is the vesicles related to the convex surface or the cis phase. This is the transfer coming from the rough endoplasmic reticulum, Name the organelle. Smooth endoplasmic reticulum. Its main function is the synthesis of the phospholipids and steroid hormones, or detoxification, or anything from the functions. Arrows point to the cisternae. Identify the organelle, rough endoplasmic reticulum. Arrows point to ribosomes, what is its function? Protein synthesis by the ribosomes. The organelle is called centriole. The red arrow points to microtubules arranged as 9 triplets. This organelle is called shaft of cilia. The yellow arrow points to the doublets of microtubules. You should mention the two words. Blue arrow points to singlets of microtubules. And if you have an arrow here, it is the cell membrane. And if you have an arrow on the fuzzy layer here, it is the cell coat. Blue arrow points to the nucleolus. Its function is the formation of ribosomal RNA. Red arrow points to nuclear envelope. If you have an arrow on these dark areas, heterochromatin, and in these light areas, euchromatin. Of course, you can see some mitochondria in the cytoplasm outside, as well as lysosome. Green arrow points to euchromatin, red arrow points to heterochromatin. If you are asked what is the type of this nucleus, it is a heterochromatic nucleus because it is containing a lot of heterochromatin and the nucleolus is not evident.